Assalamu alaikum wa rahmatullahi wa barakatuh. This is Noor Sherif from year 2, and today in this video I'm going to be simplifying the lymphocyte development lecture. I'm going to be talking mainly about humoral immunity, comparing the origin and maturation of both B and T cells, understanding and explaining the role of primary lymphoid organs in the lymphocyte development, comparing positive and negative selection, understanding gene rearrangement of TCR and BCR. But before that, we need to remember that having a single T cell will be dedicated to each epitope. Epitope is basically when you have an antigen, for example, a pathogen, and this is the area of the pathogen that is basically going to bind to our T cell or B cell or even the antibodies. So when we say that each single T cell is dedicated to a specific epitope range, we mean that it is specific. Okay, now this epitope itself, it has around of 10 to 12 amino acids, which means if we have any slight change in any of the amino acids, this is going to cause mutations. For example, the influenza virus, it has many different variants, and this is due to the mutation of its epitope, which makes our T or B cells or even the immunoglobulin lose the recognition of the epitope, which explains why some variants of influenza virus are stronger than the other. It can, our immune system is going to respond differently to it. And I'm pretty sure you guys know what is repertoire, but for whoever doesn't know, it's basically a wide range of epitope selection. So how do we have millions of T cells specific for each range of epitope? Due to positive and negative selection. Positive selection gives us the selectivity and specificity of each T and B cell, and ignoring self, which is a result of negative selection. Now, the difference between primary lymphatic organs and secondary is that primary is the location where they arise from. So we have bone marrow and thymus, and secondary lymphatic organs include the spleen, lymph nodes, and mucosa-associated lymphatic tissue. These are pure memorization. And we already know them from foundation year. We have galt, nalt, balt, and also the tonsils. And here you can notice that basically the primary lymphoid organs, they are the site at which lymphocytes form and mature. We're gonna see that in the next slide. Now, both of them, they arise from hematopoietic stem cells in the bone marrow. So we say that the location of the formation of B and T lymphocytes is the same, which is the bone marrow. Now, B lymphocytes, they remain within the bone marrow during the process of maturation, while T lymphocytes, they run away and they go to the thymus because they don't want to stay. Now, what is most important that you need to know about secondary lymphatic organs, it's basically where antigen presentation occurs. So this is when the cell starts being not naive. It would be already knows how to fight against other antigens. It's also the site of differentiation of mature naive lymphocytes into effector cells. Now, there is a difference between maturation and a cell being naive or not. So we'll go step by step. For B cells, they arise from the bone marrow, hematopoietic stem cells, and they start their maturation and they leave as immature. But where do they continue their maturation? They continue in the secondary lymphatic organs. So when they leave the secondary lymphatic organs, they're mature, but they're still naive. Okay? Which means it's like I'm comparing it to a baby that knows how to walk, how to eat, but they're still naive. They still don't know anything about life. Now, T cell, it also starts in the bone marrow, but you can notice that it completes maturation in the thymus. And remember, thymus is a primary lymphoid organ. So we can add another point, that B cell completes its maturation in a secondary lymphatic organ, while T cell completes in the primary, but in two different organs. The first one is in bone marrow and then thymus, while B cell uses both the primary and the secondary. And as a result, both of them, they're going to leave the lymphoid organs as mature and naive. They still don't know how to fight. They are still not exposed to any antigens. So this is where we need thymic education. Now, what is the purpose of this? We are going to generate randomly a diverse pool of specific T cells for each antigen. So this is related to positive selection, ensuring that self-reacting T cells are not produced. If they are produced, we're going to kill them through negative selection. Now the thymus. This is mainly observation. This is histology. So you can see that this is the cortex, which is the outer part, and this is the medulla. Now, a way so that you can remember how much amount of cells is cortex is the same. You can remember cortex by crowded. So as you can see that the cortex is mainly darker than the medulla, which means I always want you to correlate that the darker the region is, that means it has more stuff in it. So what are the stuff that is related to the thymus? More T lymphocytes. So cortex, crowded, it has more lymphocytes. But which type, immature or mature? The immature ones. All right? And we have another name for them, which is thymocytes. Now the medulla, it's way lighter, which will make you remember that it has minimal what amount of T cells. And which ones? Are they immature or mature? No, they're mature. So you can notice they're completely opposite. Dark, light. Densely packed, fewer. Immature, and it's mature. Now what is the function of the thymus? As we said, it completes the maturation and formation of T lymphocytes. And how does it go? It goes from the cortex to the medulla. Now, human thymus across the lifespan. I want you to remember 2 points. Actually 3. The first one is that thymus starts being formed at the 8th and 10th week of gestation. Alright? And in neonates, which is basically newborns, the thymus is the largest and most active. As we start to age, it starts atrophying, it starts shrinking and becomes inactive, and then it's going to be replaced by fatty tissue. Why do we have TCR before exposure? You might ask this question, and why do we have a wide range of epitopes and our body can recognize before we're even exposed to the disease? It's because early on, our own body, it does random generation, okay? And in this case, it will give us the ability to have a wide range of repertoire. And within this wide range of repertoire, we're going to have cells that are self-reactive. What am I trying to say here is that basically I'm making as much as possible cells. Some of them are going to fight this pathogen and some of them are bad. They're going to fight me. That's why I'm making this step. How do I make use of this ability? It's by basically eliminating the ones that are going to harm me. And this is using negative selection. Now, the thymus, the hematopoietic stem cells, which are basically the T lymphocytes, they migrate to thymus from 8th week of gestation, which is basically what I just said here. Now, maturation of T cells involves TCR rearrangement and expression of surface molecules, which are basically what? CD4 and CD8. CD4 for helper T cell and CD8 for the killer. Okay? Now, thymus creates environment for T cell maturation or education using positive and negative selection. And mature naive T cells, they migrate later on to peripheral lymphoid organs. So, what I'm discussing here is basically what I just said here. But the only difference is that I said after the mature naive T cells, they finish their maturation and the thymus, they're going to go to the peripheral, or we say the secondary lymphoid organs, to complete their education. We don't want them to be naive anymore. And the T cell repertoire is complete at puberty. This slide might look confusing, but I just want you to focus on one step, which is basically DN3 changing to DN4. And this basically signals what the TCR rearrangement completion. What if I go backward? Exactly happened. Basically, I had a hematopoietic precursor that enters the medulla. This is important for you to know. So it enters the medulla, but it then migrates to the inner cortex. Okay? Now, when it migrates to the inner cortex, it will be called a lymphoid progenerator, it came from the bone marrow. Now, this double negative 1 is going to change to double negative 2, and then double negative 2 to 3, and 3 to 4. And this is the most important part. And remember, all of this is in the thymus, so these T cells are going to exit as mature naive. Okay, and they exit which part? Through what exactly? Through the high endothelial venules. Now, BCR and TCR. 2 points that are common in both of them is that both of them have a variable and a constant region. Okay, as you can see, variable, constant. And both of them, their chains are linked by a disulfide bridge, this type of bond. Okay, here is it. Now, when we go to BCR, we're going to notice that we have basically 2 identical heavy chains and 2 identical light chains. Which one is the light and which one is the heavy? This one is the light chain, as you can notice, and this one is the heavy. So the bottom ones are the heavy, and the ones that are here, as if they're arms, those are the light chain. Now, you can see here also, this is the antigen binding site. So we can see that also this is where the receptors rearrangement occurs, and it is the site of the epitope binding, because again, epitope is the site or the region that is on the antigen that will bind to the BCR or TCR. Okay, now when we go back to the TCR, we also can notice here in the BCR that we have basically, we call it heavy and light. But in TCR, we don't say heavy and light. We have other terms. We call it alpha and beta chain. Okay, now it consists of one alpha and one beta linked by a disulfide bridge. Again, you can see that the variable region is the region where the antigen is gonna bind. So in both cases, both of them linked by disulfide, both of them have constant and variable. Both of them, the variable region is the region where the antigen is gonna bind. This is all you need to know. Now, TCR rearrangement. So surrogate light chain is just a temporary alpha chain, and it is used to test beta chain before moving to make the real alpha. So how is this, like, how can I understand this? Is that basically, I did explain here that I have one alpha and one beta, right? And you can see beta is a bit shorter, and even the site itself, it's quite different. Now, before you want to make the alpha chain, alpha wants to test beta. It wants to see, is it the right chain? Does it have any mistakes, any errors, any mutations, any of that? So we're gonna have something that is called surrogate light chain, which is this one, pre-TA. Okay, once it sees that the beta chain passes the test, it's fine, there are no errors, it's perfectly fine, it moves to make the real alpha chain, which is the variable region A and constant region A. I always want you to remember that the region that binds to the lymphocyte itself is the constant region, as you can see here. Okay, the constant region. And always the site that binds to the antigen is the variable region. Regarding the TCR rearrangement, so at double -3, the stage that is in the picture, as you can see, the one that I told you that is most important. At this point, basically what happens is that the T cell rearranges the beta chain, and if they see that it is successful and passes the test, it is going to express something called pre-TCR. Now, this pre-TCR basically has two stuff. The first one is pre-TA, which is this one, the surrogate light chain, and the successful beta chain. After that, we have like a checkpoint. Okay, this checkpoint will make sure, is the beta chain actually successful? It will make sure of that. So after that, if it confirms and it passes that checkpoint as well, the pre-TA is going to rearrange. So there will be rearrangement of the alpha chain to form a complete alpha-beta TCR. Now, after that, it will undergo positive and negative selection. Now, there's a difference between a pro-BT cell and pre. I can give you an example from here. It's basically DN1, DN2 is the pro, all right? And DN3, DN4 is the pre. Pro is earlier than pre, because in pro you still don't have the TCR. In pre, at least you have the beta chain and you have a surrogate alpha light chain. Now for the TCR rearrangement, so it starts at stage DN3, which I showed back here, and I said that this is the most important stuff for the TCR rearrangement. So as you remember, we said that we have what? We have the alpha chain and the beta chain. Okay, now at this stage, which is, we call it the pro TCL, which is basically the process during the making of the beta chain. All right, and I want you to keep one thing in mind, is that when I mean beta chain in T cell, this corresponds to heavy chain in B cell. Okay, so heavy in B and beta in T. But when I'm saying alpha in T cell, this corresponds to what in the B cell? It corresponds to light chain. So I'm going to be using them interchangeably. So now that I'm discussing TCR rearrangement, I'm only going to be saying beta and alpha. So at pro T cell stage, I'm rearranging the beta chain, and if it is successful, it will express the pre-TCR. طيب هلا I have the pre-TCR. What does it have? It has a successful rearranged beta chain, and it has something that is called pre-TA. Now pre-TA is basically what? A surrogate chain that we just discussed. طب شو فايدتها? It basically checks that the successfully rearranged beta chain is well or not. So at that point, we're gonna have a checkpoint أصلا يعني here. So it's gonna make sure that this one has been successfully rearranged and all is well. There's no errors, no mutation, none of that. Once it makes sure of that, it's gonna request the change of the pre-TA to an actual real alpha chain. Okay, and after that, once it changes that, we're gonna have a complete alpha beta TCR. Now they're gonna undergo يعني positive and negative selection, which we're gonna see later. Now I want you to know that same beta can pair with many different alpha chains. Okay, and the beta chain is made up of VDJ, while alpha is VJ regions, which is very, very important. Which this also means what? That the heavy chain in the B cell is going to be made of VDJ. It's going to be, the light chain is going to be made up of VJ regions in B cells. So up until this stage, we call it what? The one that has اللي هو surrogate alpha light chain. The pre-TCR is the pre-T cell, from its name, pre. And the pro-T cell is the one that is in DN1 or DN2, usually the one that is in the process of making the heavy chain, so at this stage. Okay, and now the TCR-BCR gene rearrangement. What do I benefit from this? First thing, I want you to know the germline DNA, which is basically we are born with it. Germline DNA, we're going to have extensive VDJ regions or segments. Now, from here, when we're born with this, alright? Later on, we're going to have somatic recombination, which is basically, it's like I'm choosing, yani for example, I'll give you an example. I have 3 shirts and I have 3 pants, all with different colors. I can make a combination as much as I want. Okay, but here we're gonna apply it to the genes. So for example, I want V1, D1, J1, and C. This is one match, one outfit. And then I can do another outfit, which is V2, D3, and J5, for example, and C. And I can make as many combinations as possible because I have so many genes. I need you to know that this step happens at DNA level. It is irreversible and it is random. Okay? يعني those thousands regions of VDJ, they randomly combine together and then we cut them. يعني it happens in our body at DNA level, it's cut and then they're joined together by ligases. After that, they're transcribed to mRNA because again, we're talking genes. So you're going to transcribe them to mRNA through transcription process and then translation. So we're going to translate them to proteins, which are going to form the TCR and BCR because these receptors, أصلاً, they're purely what? They're proteins. طب أنا what do I benefit from this? For example, let's say I have 2 different people, but both of them have the same VDJ rearrangement. Are they going to be the same? No. Why? Because they have different germline DNA. But what is the benefit of the somatic recombination in general? Is that so that I can have unique TCR and BCR. So we're talking in terms of affinity of the receptor itself and its recognition to other antigens. And I want you to know that this gene rearrangement happens to both the T cells and the B cells. BCR rearrangement. So we have pro-B, again rearranging the heavy chain, VDJ, and pre-B is testing the heavy with a surrogate light chain, then rearranging the light chain. So this is basically the same as what I said here. But in terms of here, we're talking about the light chain, and since it is light chain, we only talk about the VJ instead of VDJ. So as you can see, that the heavy chain, VDJ. Light chain, only VJ. So pro-B already has the heavy chain. Pre-B has the heavy chain, but it's testing what? It's testing the heavy with a surrogate light chain. If it passed this test, it's gonna change the surrogate light chain into what? Into a real light chain.
Hello everyone, my name is Abdulla Sherif. I'm going to be explaining the last part of B cell receptor rearrangement. Previously, my sister Noor explained the previous parts, which are identical to the T cells. However, there are some differences which are to be noticed. For example, the T cells, they follow a strict rules or strict steps known as double negative 1, 2, 3, and 4. The doctor mentioned that step 3 is the most important. Did not explain why, just stated, so keep it in mind. So, as my sister explained, the B cells follow the same as the T cells by doing the splicing by the RAG, fixing up by Artemius, and the enzyme TDT to increase diversity. However, the difference lies in the last step and in a new region found only in the B cells, not in the T. It's known as the constant region or the C region. This region is very important in producing the main function of the B cell. The B cell's main function is to produce antibodies, since it's the humoral part of the immune system, which is through antibodies. So we need a specific part of the DNA to code for these specific immunoglobulins. So this is the explanation here, that all B cells are naturally IgM. So basically when they are born, they are IgM producing B cells. However, they begin to switch or isotype switching based on some signals which come from helper T cells. Which helper T cells specifically? It's mentioned here, follicular T helper cells, and they are found in the follicle of the lymph node. They secrete a cytokine known as CD40. CD40 was mentioned in the second lecture we took, however, so don't get it confused. In the previous lecture, CD40 was responsible for activating the macrophage to produce some reactive species. So make sure to keep it in mind that CD40, although it is on the helper T cell, although it binds to both macrophage and B cell, they both induce different reactions or different functions, let's say. So when the CD40, which comes from the helper T cell, binds onto the B cell, it induces the transcription of an enzyme known as activation-induced deaminase. This enzyme is responsible for creating the isotype switching. By that, the enzyme comes into a region known as S and S. It attaches to this point here and this point here, creates a sort of loop, and cuts this area completely. So as you can see here, it removes this area, and we have this as a product. However, you may say, okay, how did this create diversity in the immunoglobulins? Well, this enzyme isn't only limited to this region. It may cut as farther as it wants or as less as it wants, and this will create the specific type of immunoglobulins. However, a key point to notice is that this enzyme does not touch the VDJ region. Okay, so the VDJ region, if it remains unchanged, this means that the variable region will remain unchanged. If the variable region remains unchanged, this means, so let's give an example. If this is an IgM, it's not drawn to actually. If this IgM is specific for this bacteria, let's just call it bacterium. If I change the VDJ, this means that this part, which is, as we know, is the variable region, will change. That means that if we do change this specific immunoglobulin will no longer be specific to this. However, when we leave the VDJ region unchanged, as we usually do when we do the BCR, we focus on the constant region. That's why in the DNA, if you want to link things together, in the immunoglobulin there is a C region, and in the DNA of the B cell there is the constant region. So the constant region will play a role in the constant region of the immunoglobulin. Changes in the constant region will cause an immunoglobulin to switch, for example, from IgM into IgG or IgA, and so on. So you're saying, what is the benefit of changing the constant region? Well, let's go back to the point that if the constant region changes, the function of the immunoglobulin changes. So I think there was a table here in the PowerPoint. I'm not sure where it was. I think it was after. But it stated, yeah, it's here. Each Ig has its own function. As you notice, these Ig may be specific to the same bacteria. However, because they have a different constant region, they may act differently when they find this same bacteria. And the reason why this happens is because of this enzyme, the AID enzyme, and the changing of the constant region in the DNA. So I can have an IgG, which is specific for this bacteria, performs a different function when it reaches this bacteria, and then IgA that also reaches the same bacteria but performs a different function when it finds it.
This is basically what we explained about TCR and BCR gene rearrangement. I want you guys to know that V stands for variable, D for diversity, and J for joining. Now, the DNA rearrangement causes one copy of each type of gene segment to go in any given lymphocyte. This process will generate an enormous antibody repertoire, or TCR, although some are removed due to self-reactivity. And now, how diversity is created? Mainly of 4 steps. Inheritance germline. This is we're born with it, okay? Which is basically we have coding for the proteins of the segments, the 3 segments, the variable segment, the diversity, and the joining segment, okay? And now we're gonna have the somatic recombination, which I explained a few slides back. It involves 2 enzymes, RAG1 and 2, and erythmas. Now RAG1 and 2, this is the one that cuts. It cuts the DNA to join VDJ together. And then the other one is for repairing. It is for the joining. This somatic recombination occurs after birth, and this is very important to know. هلا احنا if we don't have this cutting from rag 1 and 2, we won't have T cells, because it basically cuts these sections so that later we can join them. Okay, now junctional diversity, we use an enzyme called terminal deoxynucleotide transferase, which stands for TDT. It basically randomly adds the nucleotides at joins. What do I mean by joins? And I have here VDJ, right? So I can do VD together, and then I can do DJ together, and then I can also make a point between V and J. So at these 3 points, I can add different nucleotides, which also gives us an idea is that what if I have like the same sequence copied twice? With this step, when I add an addition of a random nucleotide, I'll make it different. So we will not have the same TCR, we are going to have unique TCRs. Now for the random assortment of chain, which is basically the organization, we already explained this as well, is that when we have the surrogate light chain and all of that, so the same heavy/beta chain can pair with many different light or alpha chain. So in this case, we're going to have more combinations. Okay, so basically now we're going to see the thymic education. But before that, we need to know that we have different type of thymic epithelial cells. So we have the cortical one and the medullary one. From its name, you know where exactly in the thymus it is located. So if we go to the cortical one, it is basically a specialized APC, an antigen presenting cell. It has both MHC1 and also MHC2. What it does is basically it presents antigen on this MHC1 and 2, and it checks the proper binding. Okay, and once it does that, for example, here I have a double positive, okay, because we did say that we have the lymphoid progenitor, and it's converted to DN1, and then DN2, DN3, DN4, and then it is converted to double positive. By the way, for your information, when I'm speaking about double positive, that means it has regulation of both CD4 and CD8 TCR. So to be able to choose to make it only CD4 or only CD8, we have this process, the positive selection. So what happens is that the cortical thymic epithelial cell is going to have, as I said, both MHC and 1 and 2. And on the MHC 1 and 2, it's going to have an antigen. And this is the double positive cell, the same one here. So they're going to interact together. On the double positive, I have the TCR. Now the TCR is going to bind with the MHC, and depending on this how strong it binds, it's going to continue its way on to being the committed type of cell. شو يعني من هالكلام؟ يعني if it binds strongly with MHC1, it's going to commit onto being a cytotoxic T cell اللي هي CD8. طيب, what if it binds strongly more to MHC2? That means it's going to commit onto being CD4, which is helper T cell. So it all depends on the binding. يعني double positive, it has TCR as it is. It also has both for CD4 and CD8. و CTEC عندها MHC1 و 2. كأنها بتقولها اختاري اللي انت عجبك. فهو it depends on the binding ability, how strong it is, and from there we take it further on to actually committing. Another type of thymic epithelial cell, which is medullary. Main thing that I want you to know about it is that the mRNA and protein level of a gene, it's called AIR gene, it's more expressed in the medullary more than the cortical. يعني الcortical عندها بس إنه very little compared to the medullary. طيب, what is the benefit of this? Basically, this epithelial cell eliminates self-reactivity, so it's related to negative selection. طيب, is there any other cell that also does this? Yes, dendritic cells. بس إحنا هون mainly we're focusing about medullary thymic epithelial cells. طيب, هلا what about the AIR gene? Tell me more about it. The AIR gene is basically, it gives the ability to express different types of tissues. What does this mean? يعني أنا هلا when I have a T cell here, for it so that I can make it not attack any part of my body, the medullary thymic epithelial cell, it has this AIR gene. This AIR gene is gonna let basically the epithelial cell to express many different type of tissues in a very complicated way that I don't want to get into. But basically, what am I trying to say? And it's trying to get the T cell to get used to all different types of tissues in the body. For example, it will start suddenly releasing insulin, or it can suddenly start releasing myelin, as if in like related to the nervous. So it wants it to get used عشان it doesn't go and attack any type of cells. طيب هلا because of that, when people lack this air gene, they're gonna have multiple autoimmunities. So with this air gene, my body can be able to differentiate between different types of tissues. When I don't have it, I have autoimmunity. So if a person has autoimmunity, we can say what? They have a defect in this type of cell. Now, if we go back to the figure, basically I explained all of this in this part, which is related to the cortex, outer and the inner. So as I said, double positive, this is the interaction that happened here. And then let's say it recognized MHC1, so it's committed CD8. And let's focus here a bit. Now it entered the corticomedullary junction. And it entered the medulla. Here we have the second checkpoint. So in here we're going to have the negative selection mainly, okay? So we're going to see any cell that reacts to self, it's going to be eliminated. So as you can see, a medullary epithelial cell or dendritic cell, it's going to kill it by apoptosis. طيب, the same thing also applies to CD4. So MHC class 2 recognized, CD4 committed, enters again the same junction and then enters the medulla. And then the medullary is like a security guard. It views it, it sees, okay, you're going to attack me, خلاص, I'm going to cancel you from the beginning. So it cancels it out and then they immigrate to the periphery. Now this is one of the most important slides. Basically the interaction that I described here is related to this, but we're going to be talking about the binding sites. So here I have MHC class 1 and here I have TCR. You can notice that we have 1, 2, 3 binding sites. Now the TCR itself with the MHC, we have 2 spots, here and here. طيب, what about the antigen that is within this MHC groove? It is going to contact with the TCR. So we have total 3 points of contact. 2 with the MHC and the TCR itself, which is related to the first checkpoint, this one, the one that I discussed here, this part. And we also have the TCR and the antigen for recognition. So all of this is related to this step. But mainly the recognition is for the antigen, because initially that is what I'm presenting. And this is what I'm testing. So for the binding itself, TCR with MHC, and then for recognition of antigen is the TCR and the antigen. Now both play a role in the positive and negative selection. And in the part that is related to the medulla here in the negative selection, and here we have the elimination of self-reactivity. Basically, we expose the cell to self-antigen. If it shows any kind of activity, then we kill it. No, we do apoptosis, so we don't give it any second chance. And for MHC to be able to bind to TCR, it needs proper binding. Please focus on these keywords. Proper binding, I need for it, okay? Proper binding means what? It is not too strong nor too weak. So it is good for detachment and reattachment. And this is the same slide. So basically what are they trying to say is that CD8 and CD4 that are here, they increase the sensitivity of the T cells towards the antigen that is within the MHC groove by 100-fold. The type of CD that T cell has a higher affinity for is kept, while the other is down-regulated. This is very, very important. And this is what I explained here. The more binding, the stronger the binding is, the affinity. That is what the cell is going to turn into. That is the commitment, يعني. طيب, for T cell to have proper binding, what should it do? It shouldn't recognize the self-antigen, because in this case it's going to attack my own body, يعني. And it should bind properly to the groove of MHC, يعني to the antigen. Okay? binding to the groove of MHC, proper affinity, outcome CD4 or CD8. طيب, what if it binds too strongly and I cannot detach it later? It's going to die. طب what if something else happens? What if it doesn't bind too strongly or proper? It doesn't even have any affinity. This is, we call it failure of positive selection, because there will be no affinity between the TCR and the MHC self-peptide. Okay, this is basically what we just mentioned, okay? Paradoxical selection. Positive selection, again, where is the location? Mostly in the cortex. It results from what? Proper binding between the TCR and MHC and self-peptide. Because, again, 2 binding spots for TCR and MHC, and then 1 binding spot for TCR and the self-peptide. طيب, what about negative selection? It's death of T cells. This mainly occurs in the medulla, which is when we have strong interaction between TCR and MHC. طب هلا let's eliminate cases. Let's say it doesn't bind proper. Let's say not strong interaction. Let's say that we remove the idea of there is no binding at all, and instead it has moderate affinity. In this case, this T cell will differentiate into regulatory T cell. When you think about regulatory T cell, I want you to think of the brake in the car. It is the brake of the immune system. It helps regulate, it prevents autoimmunity. هلا in the transition of committed double positive T cells to single positive, the cell down regulates the unwanted CD. And for example, if it binds with, as I said here, if it binds with MHC1, it's going to commit to CD8. Since it had before CD4, it's going to regulate the CD4 and upregulate the CD8. هلا not only it down regulates it also removes it from its surface. عشان احنا usually what do we say, the surface of the cell determines what it is. So if I remove this CD, I'm gonna make sure that this cell is only CD8, for example. هلا CD8 committed double positive removes CD4, CD4 committed DP removes CD8. And here is a question: which of the following is a characteristic of negative selection in T cell? Basically leads to death of T cells. Differentiation of regulatory, no. This is related to what? This is related to the moderate affinity. طيب what about proper? لا, this is positive selection. طيب it only occurs in CD8 cells? No, it's also for CD4. طيب autoimmune regulatory protein, this we talked about it. It's a transcription factor that is highly expressed in the medulla of the thymus, regulates the expression of autoantigens, and these trigger autoimmune responses. And it regulates it so that the body is exposed to it and it doesn't attack it. It facilitates the elimination of self-reactive T cells. هلا mainly here, this is the definition of each. Double negative cells, DN1 to DN4, they do not express CD4 or CD8. At stage of DN4, receptor chain rearrangement is completed, and this is, remember when I talked about TCR and BCR, resulting in a fully functional T cell receptor. DN4 to double positive, this cell transition to double positive expressing both CD4 and CD8. These double positive thymocytes are ready for selection. يعني they already have functional TCR, they have both CD4 and CD8, they're now ready for the selection process. Now DP interaction with cortical epithelial cells for negative and positive selection, mainly in the cortical it's positive. Type differentiation into CD4 or CD8, this is related to the binding that I talked about as well. Type DP interaction with medullary, this is mainly related to negative selection. So it eliminates those with strong self-peptide binding, and this is related by this transcription factor expression. Single positive cells, they're successful thymocytes. They express either, and they're ready for export to peripheral lymphoid sites so that they can face antigens. Now, B cell development. We did talk that it develops in the bone marrow. We have progenitor lymphoid cell, and then it is converted to immature B cell on too many stages. So here we have progenitor lymphoid cell. It will change to progenitor B cell, and then precursor B cell, and then immature B cell. Throughout these processes, from stage 2 till stage 3, we have heavy chain rearrangement, so it is related to VDJ. And then from stage 3 to stage 4, we have light chain rearrangement, which is related to VJ, which my brother explained. Now, this is good to know, but all I want you to know, honestly, from this is that B cell maturation is in a radial direction. Yani more mature cells are located toward the center of the bone. Remember how when we said in the thymus that we have immature cells in the cortex and we have the mature ones in the medulla? Here in the bone marrow, the one that is located toward the center of the bone is the mature cells. While the endosteal, or like the inner surface, it has a layer of the osteoblast. The B cell development occurs in the bone marrow. Light and heavy chain rearrangement occurs, leading to formation of immature B cells. These immature B cells express specific BCR. They will migrate to secondary lymphoid organs where they mature to naive and mature B cells. طيب, B cell differentiation occurs throughout life. I remember, for this point, I want you to remember the table that I explained in the beginning, where I told you in secondary lymphoid organs, it completes maturation. For the B cell selection, it's more biased than in T cells. As you remember, in T cells, we don't really give any second chances. Here in B cell, we do. So what happens if we have a strong ligation, a strong binding of the IgM, which in this case is the BCR, B cell receptor. And if we compare to T cell, هي نفس TCR. Okay? Self-antigen, this is on the MHC. And I just want you to correlate these two ideas together. But I'm speaking about B cell. If there's strong binding, we are going to arrest this B cell development. And we're going to tell it, please rearrange yourself. Rearrange the light chain specifically. So it's related at the VJ, so receptor editing. So low cell surface IgM. A new receptor specificity is now expressed. And if the new receptor is still self-reactive, then we don't give it a chance and the B cell undergoes apoptosis. If the new receptor is no longer self-reactive, the immature B cell migrates to the periphery and matures. This is a summary of the lecture. B cell mature in the bone marrow, T cell in the thymus. T cells undergo negative and positive selection to generate mature T cells. Following maturation, B and T migrate to secondary lymphoid organs so that they are not naive anymore. And gene rearrangement ensures the diversity of lymphocyte specificity. Thank you for watching, and I truly hope this video was helpful.